For my Adopt a Microbe project, I studied the bacteria D. Vibrio vulgaris. It is a gram-negative anaerobic bacteria. Pictured here are two different microscopic images of the sulfate-reducing bacteria. This shows their curved rod-shaped nature. D. Vibrio vulgaris is considered an opportunistic pathogen as it can exist in humans asymptomatically. However, it tends to only exist asymptomatically when it appears in low counts. Otherwise, this bacteria is known to cause abdominal infections such as an abdominal abscess, cholecysticitis, or ulcerative colitis in some cases. This is due to its role as a sulfate-reducing bacteria. This process produces excess hydrogen sulfide in the gut. In the majority of cases, this is a negative thing contributing to various intestinal diseases. However, a potential benefit is hydrogen sulfide's role as a signaling factor for the nervous and cardiovascular systems. In most cases, hydrogen sulfide reaches toxic levels, causing these various inflammatory bowel diseases. For these reasons, D. Vibrio vulgaris tends to be found in the feces or intestinal tract of both humans or animals. Further research may allow for scientists and physicians to determine better preventative and treatment methods for GI diseases. Although in humans there are more negative effects than positive, this sulfate-reducing bacteria may have ecological benefits. D. Vibrio vulgaris may be used for toxic metal remediation. This means that toxic metals such as mercury, arsenic, and lead can be changed to a less harmful form. This process could play a big role in reducing environmental pollution. The paper that first describes this bacteria is by John R. Postgate and L. Leon Campbell. It is called The Classification of the D. Vibrio species, the non-sporulating, sulfate-reducing bacteria, which appeared in the journal Bacteriological Reviews in 1966. This bacteria is often referred to as D. Vibrio vulgaris Hildenborough, since it was first isolated from clay in Hildenborough, England. It was discovered in the forests and grazing fields in this village. D. Vibrio means a curved, motile, anaerobic bacteria that reduces sulfates to hydrogen sulfide, and vulgaris means common. D. Vibrio vulgaris is a biosafety level 1 microbe, meaning it is at low risk. According to the CDC, this means that working with this bacteria in a laboratory setting should not cause harm to healthy adults. Working with this microbe can be done on an open lab bench. PPE should be worn and a sink for proper hand washing must be accessible. The strain designation I chose is NCIB 8303, a variation of D. Vibrio vulgaris Hildenborough. This strain comes in a freeze-dried format and must be stored in cold temperatures prior to use. The ideal medium is a modified Bars medium, as sulfate-reducing bacteria must be grown on a specialized medium. Ideally, D. Vibrio vulgaris should be grown at 30 degrees Celsius and in anaerobic conditions. D. Vibrio vulgaris is a gram-negative bacteria. It is pictured on a gram stain above. The pink color indicates that the bacteria is in fact gram-negative. This means that there is a presence of an outer membrane and that there is a thin peptidoglycan layer. Its morphology is bacillus, which can be seen by the rod-shaped cells. Scanning electron microscopy can also be used to visualize D. Vibrio vulgaris. The Hitachi S4700 field emission scanning electron microscope was used to view D. Vibrio vulgaris. It was viewed at a magnification of 6,000 and at a working distance of 5 to 6 millimeters. Reazurin was used as a redox dye. A gram-negative result would show as the D. vulgaris turning to a pink color on the plate. To test for the oxygen requirement, D. vulgaris would be placed in an oxygen chamber. Since it's anaerobic, it will not grow in the presence of oxygen. On ordinary blood agar plates, D. vulgaris results in gamma hemolysis. The SIM medium test can be used to determine motility, which D. Vibrio vulgaris tests positive for as well. Cetobacterium someri and Oscillobacter rumidium test positive for the same conditions as well. However, they additionally test positive for nitrate to nitrite and glucose fermentation, allowing D. vulgaris to be differentiated. For the blood agar test, gamma hemolysis means that there is no hemolytic activity. On the McConaughey-Agar test, this negative result means that D. salvibrio vulgaris is lactose non-fermenting. A mannitol positive test means that acid was produced, changing the phenol red broth to a yellow color. And finally, a positive yellow mannitol salt agar test means that acid has been produced from the breakdown of mannitol. To amplify my D. vulgaris sequence, I researched potential primers until I found these ones that worked. The melting temperature for the first primer is 64.8 degrees Celsius and 65.8 for the second. The GC content is about 48% for the forward primer and 44% for the reverse. When I did the NCBI blast, I was able to get D. salvibrio vulgaris. Additionally, there were a few different D. vulgaris strands and other strands in the D. salvibrio family that were in my results. Pictured here is my phylogenetic tree for the microbes studied by my classmates as well as my own. There are a variety of drugs that are effective against the D. salvibrio species. The most commonly used are clindamycin, metronidazole, chloramphenicol, and impendium. There are no other known treatment methods, nor is there a vaccine. In order to prevent or manage diseases that could be resulting from D. salvibrio vulgaris activity in the gut, is healthy eating and improving one's diet. High levels of animal proteins, saturated fats, sugar, and salt should be avoided. Additionally, there are 2 elimination diets known to reduce symptoms for those who already have GI disorders. The low FODMAP diet stands for low levels of fermentable oligo, di, monosaccharides, and polols, which are poorly absorbed carbohydrates. This elimination diet counteracts imbalances in the gut microbiome. The second elimination diet that has been deemed successful is GFD, or the gluten-free diet. A study performed showed in just 1 month the numbers of harmful gut bacteria, such as D. salvibrio vulgaris, greatly reduced by eliminating gluten from one's diet. On May 21, 2017, in the city of Durban in South Africa, a news article came out in the Independent Online regarding toxic fumes coming from D. salvibrio vulgaris in a landfill. Those who were exposed to the fumes developed allergic rhinitis and asthma symptoms. The fumes were present for several months and many citizens were complaining. Authorities and officials denied that there was anything harmful coming from the landfills. However, healthcare workers deemed this as a medical emergency. To conclude, these are my references in the order they appear within the presentation. Thanks for watching.